secondary structure, tertiary structure, and the quaternary structure. So this quaternary structure, it may not be present in all the proteins since it is present only in those proteins which have more than one subunit. Okay, more than one peptide or the polypeptide chain is present in the protein, only then the quaternary structure will be present. So if the protein is made up of only one polypeptide or only a peptide chain, then this type of the proteins will not have the secondary, the quaternary structure. Okay. Then yesterday we also discussed that what are the forces that maintain the structure of the protein. So in that, what we have said or what we have seen is that there are two types of the forces that maintains the structure of the protein. Okay, that is, one is the covalent bond, and among the covalent bonds, one is the peptide bond, and the other one is the disulfide bond. So these two forces are the covalent bonds that are present in the protein structure. Then among the non-covalent forces, the forces which maintain the protein structure, it includes the hydrogen bonding, hydrophobic interaction, ionic interaction, and the van der Waals forces. Okay, so these are the forces that maintain the structure of the proteins. Okay, now primary structure. What do we mean by the primary structure of the protein? So the primary structure of the protein, it refers to the arrangement of the proteins in the arrangement of the amino acids in the protein. So how the amino acids are placed in the linear sequence of the protein structure is called as the primary structure. Say, suppose you have the amino acids, so which amino acid will come in the first position, then which amino acid will come in the second, third, fourth, fifth. Okay, so how these different amino acids are arranged in the sequence of the protein is called as the primary structure. Okay, so primary structure, the linear sequence of the amino acids forming the backbone of the protein is called as the primary structure of the protein. Now here in this now, if you see the diagram here, okay, so first one, we know that this is the amino acid serine. Next one is the glycine. You have the tyrosine, you have the alanine, then you have the amino acid leucine. So this now arrangement of the amino acids in this protein is that first it is the serine, then glycine comes, then tyrosine, then alanine, then the leucine. So this arrangement of the amino acids in this protein is called as the primary structure. So simply, you can just tell that primary structure means it is the way in which the proteins are arranged in the sequence, in the amino acids are arranged in the sequence in the protein molecule. So that is called as the primary structure. Okay. Next is the secondary structure. Okay. So secondary structure of the proteins, it refers to the special arrangement of the amino acid residues that are near one another in the linear sequence. So that means this is how the different amino acids which are near to each other in the linear sequence, so how they are arranged in space. Okay, how these amino acids are arranged in space, it is called as the secondary structure. Okay, the conformation of the polypeptide chain by twisting or folding, it is called as the secondary structure. So generally, that arrangement of the amino acids in space, it is done by twisting or the folding of the polypeptide chain. Okay. So two types of the secondary structures are known for the proteins. One is the alpha helical structure, and second one is the beta sheet structure. So two types of the secondary structures has been identified in the protein that are called as the alpha helix, and second one is the beta sheet structure. The helical structure, the helical structure was first discovered, so the name alpha is given. Then after the helical structure, the sheet structure of the protein, it was identified, it was discovered. So comes the second, beta. So alpha and beta, it is just the sequence in which these structures are discovered or identified in the protein molecule. Yes, sir. 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 Yeah, something it is there. I'm not so sure, but within, I think, 10 or 15 days of the receipt of the material, it has to be there. But it's better to confirm from those persons who has done. Actually, for us, that rules I have gone through it, but since we are not operating it, maybe some problems, maybe it is there. So better to confirm from there. Sir. Takes. Okay. Now both these structures, that is the alpha helical structure and the beta pleated sheet structure, this was okay. So both these structures were proposed by Pauling and the Corey. Okay, it is the same team who has discovered this. Now let's come to the alpha helical structure of the protein. So the alpha helix, it is a tightly coiled packed structure. Okay, it is a tightly packed coiled structure with the amino acid side chains extending outward from the central axis. Okay, we'll just come to the structure. Then this structure is stabilized by extensive hydrogen bonding formed between the hydrogen atom attached to the peptide nitrogen and oxygen attached to the peptide carbon. So this structure, how it is stabilized? It is stabilized by the hydrogen bonding. Then hydrogen bonding, what are the groups that are involved in the hydrogen bonding? Okay, so there is hydrogen bonding, it is between the hydrogen and oxygen. Okay, so this linkage, it is between the hydrogen and the oxygen. So how it is formed? Which hydrogen? The hydrogen that is attached to the peptide nitrogen. Okay, so if you see the backbone of the protein, then you will find it that it is the amino group nitrogen, then alpha carbon, then carboxyl group carbon. Okay, then comes the amino nitrogen. So this hydrogen attached to the amino group. Okay, amino group attached to the alpha carbon. So this hydrogen attached to the nitrogen that we call it is the peptide nitrogen, and oxygen atom attached to the this one, that carbon. Okay, peptide carbon, that is the carboxyl group carbon. So these are the two elements that are attaching, that is involved in the formation of hydrogen bonding. Okay, so all the peptide bonds except the first and the last in the peptide chain participate in the hydrogen bonding. So there will be different peptide bonds. So of that, the first one and the last one is not involved in the hydrogen bonding. All the other nitrogen and that carboxyl group is involved in the hydrogen bonding. Now, each turn of the alpha helix, it contains 3.6 amino acid residues and travels a distance of 0.54 nanometer. The spacing of this amino acid is 0.15 nanometer. Okay, the alpha helix, it is a suitable conformation formed spontaneously. Okay, so this structure, it is formed spontaneously because it is thermodynamically feasible. Okay, the right-handed alpha helix is more stable than the left-handed helix. Okay, so a right-handed helix turns in the direction that the fingers of the right hand curl when its thumb points in the direction of the helix rises. Okay. Then the certain amino acids, particularly proline, it disrupts the alpha helix. It destabilizes. Okay? It destabilizes the alpha helix. So large number of the acidic amino acids. So acidic amino acids, we know that it is the aspartic acid and glutamic acid. Then the basic amino acids, so 3 amino acids are there: lysine, arginine, histidine. Then amino acids also interfere with the alpha helical structure. So what are the amino acids? That is destabilizing the alpha helical structure. So one is that the amino acid, the proline, then acidic amino acids, that is aspartic acid and glutamic acid, then basic amino acid, that is lysine, arginine, and histidine. They destabilize the alpha helical structure of the protein. Okay, see here. Now this is how the protein is formed. Okay, now you have this line. It is showing is the protein. Okay, it is the backbone of the protein. So this, it is turn around. Okay, this is turn like this to form a helical structure. Okay, then here the side chain of the amino acids, it will be buried inside. Okay, the side chain of the amino acids in the protein structure, they will be buried inside the helix. Okay, then this structure, the alpha helical structure, is this stabilized by one force, that is the hydrogen bonding. And hydrogen bonding here, it is formed, just you see it here, that this is the hydrogen attached to the peptide nitrogen. So this is forming a hydrogen bonding with the oxygen attached to the carboxyl carbon. Okay, so like that, all the hydrogen, it is formed between the hydrogen attached to the peptide nitrogen and oxygen attached to the peptide carbon. Okay, so like this will be formed. Then the other point that has been mentioned about the feature of this alpha helical structure is that this one turn of the helix. Okay, so when this peptide chain, it turns 3 degrees. Okay, 360 degrees. That is one turn of the helix. Then the distance traveled by this is so it contains 3.6 amino acids and it travels a distance of 0.54 nanometer. So here, the number of amino acids present in when this one peptide chain turns by 360 degree, it is 3.6. So 3.6 amino acids will be present in one turn of the helix, and the distance travel is 0.54 nanometer. Okay, so distance from this point to this point, it will be 0.54 nanometer. Okay, so the second structure, it is the beta pleated sheet. So this is the second type of the structure proposed by Pauling and Corey, hence beta after the alpha. Okay, so it is composed of 2 or more segments of the fully extended peptide. Here, the fully peptide chains, they are lying one above the other as a sheet. In the beta sheets, the hydrogen bonds are formed between the neighboring segments of the polypeptide chain. In the alpha helix, it is in the same chain, okay, but here it is between the neighboring chains. Okay, see here. So this is one chain. This is the other chain. Okay, one chain is here, then second chain it is here. So like that, the different chains, they are forming a sheet structure, and the structure is maintained again by the hydrogen bonding. The hydrogen bonding is same. It is the hydrogen attached to the nitrogen of the one chain with the oxygen attached to the peptide carbon of the another chain. So here the hydrogen bonding is between the neighboring peptide chains. Okay, but in case of the alpha helix, it was in the same chain, and that is forming the coil structure. Here it is the sheet structure. Okay, next is the tertiary structure of the proteins. So the tertiary structure of the proteins, it is also just the arrangement of the groups in space, similar to that of the secondary structure. The difference here is that it considers the amino acids which are far apart in the linear sequence. In case of the secondary structure, it was those amino acids which are near to each other, but in case of the tertiary structure, it considers the amino acids which are far apart in the linear sequence. Okay, so it refers to the spatial arrangement of the amino acid residues that are far apart in the linear sequence and to the pattern of the disulfide bond. So one more addition here, it is that it also considers the disulfide bonds present in the protein. The tertiary structure, it is a compact structure with the hydrophobic side chains held interior. So the hydrophobic side chain, some of the amino acid, it has the hydrophobic side chain. So that hydrophobic side chains will be buried inside the molecule. Okay, while the okay, so this type of the arrangement ensures stability of the molecule. Then lastly, the structure of the protein that are maybe present in the proteins, it is the quaternary structure. So proteins containing more than one polypeptide chain, it exhibit an additional level of structural organization. Okay, so that means this type of the structure, the quaternary structure of the protein, will be present only in those proteins which has more than one polypeptide chain. Okay, so its polypeptide chain in such a protein is called a subunit, and quaternary structure refers to the spatial arrangement, the arrangement in space of the subunit and the nature of their context. So how the different subunits are arranged in space and how the different subunits, they are in contact. So that is referred to as the quaternary structure. So for example, hemoglobin, it consists of 2 alpha and 2 beta chains. It has the 4 subunits. So of that, 2 are of the alpha chain, and then other 2 is the beta chains. The arrangement of these subunits, that is the 2 alpha and the 2 beta chains in space, is referred to as a quaternary structure. So that means it is simply the arrangement of the subunits in its space in the protein molecule that is called as the quaternary structure of the protein. Okay. Now, coming to the classification of the proteins. So, functional, again, in the case of the amino acids, we have seen that the amino acids may be classified into based on their structure, on their solubility, then based on their nutritional status, and then all that. Okay, metabolic fat. So there was the different ways to classify the amino acids. Similarly, the proteins also can be classified in different ways. Okay, so one of them is the functional classification of proteins. So based on that, what type of function is performed by the proteins are divided or classified into many groups. Okay, so based on the function performed by the proteins are classified into the following groups as structural proteins. So some of the proteins, they are the structural component of the living organism. Say, suppose keratin, the keratin of the hair and the nails, collagen of the bone. So they are the structural component. Okay, so they will be classified as the structural proteins. Then some of the enzymes, some of the proteins, it has the catalytic activity. They have the enzymatic activity, so they are classified as enzymes or catalytic protein. So many enzymes will be discussing it when we discuss the metabolism. Then some of the proteins that are involved in the transportation of the molecule, like say hemoglobin. Hemoglobin is involved in the transportation of the oxygen and the carbon dioxide. Say serum albumin, it is involved in transportation of the fats, lipids, and all that. Okay, so if the proteins are involved in the transportation of the molecule, they are classified as the transport proteins. Then some proteins, they act as the hormone, so that will be classified as the hormone proteins. Example is the insulin, growth hormone, glucagon. There are so many. Okay. And contractile proteins, say the muscle contraction, it requires the proteins. Okay. So example is the actin and myosin. So these proteins which are involved in the muscle contraction and all that, they will be considered. They will be classified as the contractile proteins. Then some proteins, they are present as a storage form of the protein. Example is the albumin, okay, glutein. Then genetic proteins. So you have heard about the nucleoproteins, which are found in association with the DNAs. Okay, so that are called as the genetic proteins. Defense proteins like the hemoglobin, say IgG, IgM, IgA, and all that. Okay, all the immunoglobulins that are involved in the defense mechanism. Okay, so they are classified as the defense proteins. Okay, then receptor proteins, some of the proteins, they are present as a receptor of the hormones. Okay, so like that, based on that, what type of function is performed by the proteins, it may be classified into many groups. Okay, so these are the groups, that is the structural proteins, enzymes, transport proteins, hormone proteins, contractile proteins, storage protein, and so on. Now, based on the nutritional status, okay, the proteins are also classified. So that are the nutritional classification of the proteins. So one is the complete proteins. Complete means these proteins have all the essential amino acids. So earlier we have seen that there are 10 amino acids which are essential. Okay, so if the protein contains all the 10 essential amino acids in sufficient amount, then that will be classified as the complete proteins. Okay, so these proteins have all the 10 essential amino acids in the required proportion by the body to promote good health. Okay, so example of this is the egg albumin, the milk casein. Okay, so these are some of the proteins which has all the essential amino acids in sufficient quantity. Then partially incomplete. So here they partially lack one or more essential amino acids and hence can promote a moderate growth. That means this type of the proteins, they contain the essential amino acids, but some of the amino acids present, the amount of the some essential amino acids may not be sufficient. Okay, it is not present in sufficient amount, so it is called as the partially incomplete protein. Example is the wheat and the rice proteins. Okay, so they contain the limited amount of the lysine and the tyrosine. Okay, next is the incomplete proteins. So these proteins completely lack one or more essential amino acids. Okay, so one or more essential amino acids is not present at all in this type of the protein. So that means they are completely lacking one or two essential amino acids. Hence, they do not promote the growth at all. Okay, example is the gelatin, which lacks the tyrosine, then zinc, which lacks the tryptophan and the lysine. Okay, then protein classification based on the chemical nature and solubility. Okay. So this is a more comprehensive and a popular classification of protein. So there are different ways to classify it. The most comprehensive and popular classification of the protein, it is based on their chemical nature and the solubility. Okay. So it is based on the amino acid composition of the protein, structure, shape, and the solubility of the proteins. So proteins are broadly classified into three major groups under this. Okay, based on their chemical nature and the solubility, the proteins are broadly classified into three major groups. One is the simple proteins. Okay, composed of only amino acid residues. Next is the conjugated proteins, means it contains some other additional groups in addition to the amino acids. So besides the amino acids, these proteins contain a non-protein moiety, non-protein part, known as prosthetic group or the conjugating group. So conjugated proteins, it contains the additional groups. Additional groups means the groups other than the amino acids. So that additional group, it is called as the prosthetic group or the conjugating group. Okay. The third one, it is the derived proteins. The third one is the derived proteins. So these are the denatured. Hello? Hello? Yeah. Yeah. That is okay. Please start. I'm coming. I'll come within 5 minutes. I'll come. Okay, so these are the denatured or degraded proteins of the simple and the conjugated proteins. So in the case of the lipids, we have seen that when the simple lipids and the compound lipids, when they are degraded, okay, then that part, that the degraded product, if they have the properties of the lipids, we call it as the derived lipids. So like that, when these simple proteins and the conjugated proteins, when they are degraded, the product form, we are classifying as the derived proteins. Okay, let's stop here. I have to go and attend some bhaiva, so we'll stop here. We have another, I think, 5 more slides. So we'll do in the next class. We'll discuss about that. And if you have some more questions or this one, then you go through it. Along with that, we'll discuss the remaining the slides also. I think I have already shared this PowerPoint slides. This one, I'll check it. If I have not shared it, I'll share it today. Okay, so you can just go through the slides that I'm sharing it. And so, if any difficulty or if something is not clear to you, then we can discuss that topics along with the portion that which we have not covered today. Okay, so let's stop here for today. Okay. Called as a simple amino acid means these are the proteins which contains only the amino acid. It doesn't have the other additional groups. Okay. Now the simple proteins again it may be of two types. So one is known as the globular protein, another one is the fibrous protein. So again the simple proteins it is classified into two, as globular proteins and the fibrous proteins. The globular proteins these are spherical or oval in shape. Okay. So part they are present. Say if it is present in the blood, we call it as the serum albumin. If it is present in the egg, we call it as the alpha albumin. If it is present in the milk, we call it as the lacto albumin. So different types again of the albumin based on that in which where they are present. Either it is in the blood, it is in the egg, or it is in the milk. Okay. Then the second example is of the globulins. So one is that which is present in serum or in the blood, that is the serum globulin. Then gluteins like the glutein in wheat, gene in the rice. Then histones, these are the basic proteins soluble in water, and dilute acids, they are present in the nucleic acids. Okay. Then globulins, protamines. Okay. So like that, the globular proteins, again, it is of the different types. So some of the globular proteins, the examples of the globular proteins are albumins, globulin, gluteins, prolamins, histones, globins, and the protamins. Okay. Then the other group is the fibrous protein. So they are fiber in shape. Okay, they are fiber in shape, and they are insoluble in water, and they are resistant in the digestion. Means these are those proteins which contain some additional groups in addition to the amino acids, and there are many types based on that what type of group is present in the protein. Okay? So that group which are present in the protein we generally refer to as the prosthetic group. Okay? They are the prosthetic group. So the conjugated proteins are those proteins which contains the prosthetic groups. Okay? So based on the groups that are present, it may be called as the nucleoproteins, means containing the nucleic acid. Nucleo, it comes from the nucleic acid. So these are the proteins which contains the nucleic acid as the prosthetic group. Glycoproteins, so glyco, we know that it refers to the carbohydrates. So these are the proteins which contains the some amount of the carbohydrates. Lipoproteins, so these are the proteins which contains the lipids. Phosphoprotein, so when the prosthetic group is the phosphate. If the phosphate or the phosphoric acid is present, we call it as the phosphoproteins. Chromoproteins, chromo, we know that it is color. Okay, so that means it contains some groups which gives the color, like say hemoglobin. In case of the hemoglobin, in addition to the amino acids, the peptide chain, the heme group is present which gives the color. Okay, so it is a type of the chromoproteins. Then you have the cytochromes where the heme group is also again present. Then metalloproteins means containing the metal ions. Okay, like say ceruloplasmin which contains the copper, carbonic anhydrase which contains the zinc. Okay, so that means these are the proteins which contains the metal ion. So based on that what type of prosthetic group is present, what type of additional group is present, the conjugated proteins, it may be classified into many, like say nucleoproteins, glycoproteins, lipoproteins, phosphoproteins, chromoproteins, metalloproteins. Okay, so it is all, the name is given on that what type of prosthetic group or the additional group is present in the protein molecule. Okay, the third group that we have given, it is the direct proteins. Okay, so direct proteins again are of two types. One is the primary derived, and second one is the secondary derived. So the derived proteins, again, it is further divided into two as the primary derived proteins and the secondary derived proteins. So these primary derived proteins, these are the denatured or coagulated or the first hydrolyzed products of the protein. So when the protein is denatured, there is no much change, but it lost the native conformation. Okay, same thing, it is the coagulation, so it is a semi-solid form that is formed due to the denaturation of the protein. Or when the protein is hydrolyzed, okay, so the first product form on hydrolysis of the proteins, it is also called as the primary derived proteins. Okay. So in primary derived proteins, what are the proteins that are coming? So these are the denatured proteins or the coagulated proteins, or the product form just on the initial hydrolysis of the proteins. Okay. Then comes the secondary derived proteins. That is the second group among the derived proteins. It is the secondary derived proteins. So these are formed on the progressive hydrolysis of the protein. So when the proteins, when it is hydrolyzed, the initial product form, it is the primary derived. But if the protein is allowed to be degraded further, then the product form will be the secondary derived proteins. Okay. So progressive hydrolysis, products of the protein hydrolysis, and that products is called as the proteases, peptones, polypeptides, and the peptides. So these are the products that will be formed on the progressive hydrolysis of the proteins. The primary derived proteins, so it includes the coagulated proteins, the proteins, the earliest products of the protein hydrolysis by the enzymes, dilute acids, alkalis, etc. Example is the fibrin formed from the fibrinogen. So we know that in the blood clotting, the fibrinogen is hydrolyzed. Okay, some portion of the fibrinogen is removed to form the fibrin. So that fibrin, it forms the blood clot. Okay, so that is a primary derived protein. The protein itself, it is the fibrinogen, then it is converted to the fibrin. Okay, metaboproteins. So second stage metaproteins. So it is the second stage products of the protein hydrolysis obtained by treatment with slightly stronger acids and alkalis. That is acid and alkali metalloproteins. Okay. Next is the denaturation of the protein. So what do we mean by the denaturation? So denaturation means denaturation means it is the native conformation of the protein is changed. So that is called as the denaturation. So this is defined as the phenomenon of disorganization of the native protein structure. So it is called as the denaturation. So that means the native conformation, when the protein is in its natural form, it has its own structure. Okay, its own structure. So if that native conformation, the native structure is destroyed, then that is called as the denaturation. Now this denaturation, it results in the loss of the secondary, tertiary, and quaternary structure of the proteins. Okay, so here, what the changes it occurs? The secondary, tertiary, and quaternary structure of the protein is destroyed. So this involves a change in their physical, chemical, and biological properties of the protein, because the proteins' physical property means they are soluble. In the native conformation, some of the proteins it may be soluble in water. But when their native conformation is changed, when their structure is destroyed, then the proteins which were soluble initially or in its native conformation, it may not be soluble now. It may become insoluble in the solvent in which it was soluble. Okay, so that means it changed in the properties, the physical properties. Okay, like that, it will lead to the change in their chemical properties, and it will lose the biological properties. The biological properties of the proteins is present only when it is in the native conformation, or you can say that the proteins they can perform their function or their activity only when they are in the native conformation. So if the native conformation is destroyed, if it is changed, then these proteins will lose the biological activity. Okay, the agents which denatures the protein, It may be of the physical, like the heat, violent shocking, X-rays, UV radiation, etc., or it may be chemicals like the addition of the acid, alkalis, organic solvents, and all that. Okay? Even the salts, okay? Salts of the heavy metals. So all this it may denature the native conformation of the protein. Okay, so those agents which destroys the native conformation of the protein, it may be physical, it may be chemical. Okay, now as we have mentioned that the denaturation of the protein, it is the change in their physical, chemical, and biological activity. So let's see what are the characteristics of the denaturation. What changes it may occur. Okay. The native helical structure of the protein is lost, but the peptide bonds are not destroyed. So we know that the secondary, tertiary, and quaternary. So this is just the arrangement of the groups in the protein in their space. Okay. So this how these groups in the protein will be arranged in a space that will be lost. Okay. So that means the secondary, tertiary, and quaternary structure is lost. So that forces which are maintaining that secondary, tertiary, and quaternary structure is destroyed. So we know that the forces which are involved in maintaining the secondary, tertiary, and quaternary structure is the non-covalent forces. Okay, non-covalent forces, and it is mainly the hydrogen bonding, van der Waals forces, ionic interaction, and hydrophobic interaction. So these four types of the interaction is destroyed when the protein is denatured. But the primary structure, in the primary structure, we know that amino acids are arranged in the sequence, and these different amino acids are held together by the peptide or the amide linkages. So this is not destroyed. The primary structure of the protein is not destroyed, but the secondary, tertiary, and quaternary structure of the protein is destroyed on denaturation. Okay, one is that. Next is that the protein loses its biological activity, say like the enzymes. So the enzymes, they will perform their active function only when they are in native conformation. But if the enzymes are denatured, then they cannot perform their activity. Means the biological activity is lost. Okay. The denatured proteins becomes insoluble in solvent in which it was originally soluble. So that is what I just mentioned it when we discussed the previous slide, that these proteins it may become insoluble when they are denatured. Viscosity of the denatured protein solution increases while its surface tension is decreases, because the proteins it becomes fibrous now. Okay. Instead of the globular one, it may become the fibrous. So that increases the viscosity of the protein solution. Denaturation is associated with the increase in ionized and sulfhydryl group of proteins. Okay. So when the proteins they are in a native conformation, then the that should be exposed. Denatured protein is more easily digested because now it is in the extended form. It is in the fibrous form, so it will be easy for the enzymes to go and attack the peptide bond. Okay. So because of that, the denatured proteins they are easily digested. This is the reason that why we use meat, fish, and all that. Okay. So when it is cooked, the proteins will be denatured. So it is easily digested by the enzymes which are present in our system. So cooking causes the protein denaturation, and therefore cooked proteins are more easily digested. Okay. Denaturation may be reversible or irreversible. Okay. So some of the denaturation of the protein may be reversible, then some are irreversible. So suppose when you boil the egg, okay. When you boil the egg, then it becomes a solid. So that is simply a denaturation of the protein, and which is irreversible. You cannot revert back. The boiled protein, you cannot bring back to its form, the liquid form. once it is denatured. So that is irreversible. Say, but some of the proteins which are present in the solution, you may add some salts like the sodium ammonium chloride. Okay? You add the ammonium chloride, then the proteins will be denatured and you will get as a precipitate. But once, so that is denaturation. So you can remove the salts that you have added, that is the ammonium chloride, that is ammonium sulfate which you have added. It can be removed from the protein, then the protein will become again soluble. It will be dissolved in the solution. That means that denaturation can be reversed. Okay, so like that, some of the denaturation for some of the proteins, that the denaturation may be reversible, but for some of the proteins it is irreversible. So this denaturation process, it may be reversible or it may be irreversible. Okay, the denatured proteins, it cannot be crystallized. Then there are some more terms related to the denaturation of the protein, that is called the coagulation and the flocculation of the proteins. The coagulation of the proteins and the flocculation of the proteins. The term coagulation, it refers to the semi-solid viscous precipitate of the protein. So when the protein is denatured to form a semi-solid viscous precipitate, then we call it as the coagulation. So irreversible denaturation of the proteins results in the coagulation. I have just given you an example of boiling of the egg. So the egg, the liquid form, when you boil it, is converted to the solid. So this is the coagulation. Then the flocculation is the process of protein precipitation at isolated pH. So when while discussing the amino acids, we have defined it as the isolation point. Okay. So like that, since the proteins are also the polymers of the amino acids, the different proteins, they will also have the isolated pH. Means the pH at which this protein molecule will have the non-net charges. Okay. So at the isolated point, the proteins are precipitated. They will not be soluble, but to be precipitated out. Okay. The example is the formation of the what we call so the milk protein, in the milk, the protein casein is present. Okay? So that casein, we can adjust the pH of the milk to 4.6. Okay? So at 4.6 is the pH of the isolated one of the casein. So when we adjust the pH of the milk at 4.6, then the protein casein will be precipitated out from the milk. Okay? So you can just separate out the casein. So flocculation, it is the process of protein precipitation at the isolated pH. Then the precipitate is referred to as the flocculum. So example is that milk protein casein can be easily precipitated by adjusting the pH to its isolated pH, that is 4.6, by the dilute acetic acid. Okay, so this is what we are making paneer. So paneer is how we make the paneer. So simply we add, say, lemon. Okay, we add the lemon, or we may add the acetic acid, just to bring down the pH of the milk to 4.6. So when the milk pH is 4.6, since the 4.6 is the isolated pH of the casein will be precipitated out. Okay? That is, we are removing as the paneer. Okay? So like that, by adjusting the protein to its isolated pH, we can precipitate out the proteins. So flocculation is a reversible process. So here you have just brought down the pH of the milk to 4.6. Now you can increase it. So when you bring down to 4.6, the casein is precipitated out. Now to that you can increase the pH. Okay? You can increase the pH. If you bring back the milk pH to 7, then again this milk, the casein will be dissolved in the milk again. Okay? So that means this process, the precipitation of the casein, it can be reversible. Okay? So like that, the flocculation is a reversible process. Then The proteins, it can also be precipitated by many other techniques. Okay. One is that the proteins exist in the colloidal solution due to the hydration of the colloid. So how the proteins are dissolved in the solution is just the hydration. Okay. And this hydration is because the groups which are present in the protein molecule, they can form hydrogen bonding to water. So they are soluble in water. Now, the proteins it can be precipitated by dehydration or neutralization of the polar group. So, if this water is removed from by, say, if you can remove it by the process of dehydration, then protein will be precipitated out. Okay? Then other one is that just that polar groups which are forming the hydrogen bonding with the water. So, if you can neutralize that, okay, if you can disrupt the hydrogen bonding between the water molecule and the groups which are involved in the protein, then they will be precipitated out from the solution. Okay, then precipitation of the protein at the isolated point, that is the example of the casing that we have just discussed it. Then precipitation by the neutral salts. So the neutral salts like the ammonium sulfate or the sodium sulfate, they can neutralize the groups. Okay? They can neutralize the groups in the protein molecule so that they can be precipitated out from the solution. Okay? Precipitation by salts of the heavy metals. So heavy metals, the salts of the heavy metals, like say the lead, mercury, and then iron, zinc, cadmium. So it also causes the precipitation of the proteins. So these metals being positively charged added to the protein solution, which is the negatively charged in alkyl medium, results in the precipitation formation. So this also, when it is added, then it makes changes in the interactions that are going on in the protein molecule, which results in the destruction of that interaction, and that results in the precipitation of the proteins. Precipitation by anionic or alkaloid reagents, say, for example, the TCA, tricarboxylic acid, then sulfoxylic acid, phosphotungstic acid, citric acid, acetic acid, phosphomolybdenic acid. So all these, if you add it to the protein solution, they also lead to the precipitation of the proteins. And some of the organic solvents, it also precipitates. So this is all about the amino acids and proteins which we have to discuss it. And with this, we have already finished the unit 1. Okay? And the unit 1, what we discuss is biomolecules. Okay? So here we started with the scope of the biochemistry. Then we discussed about the biological membranes, then acid base, then buffer system, then we have discussed the carbohydrates, lipids, nucleic acids, proteins, and amino acids. Okay? So all that constitutes the first unit, and we have completed the first unit. Now we shall start with the second unit. Okay? The first topic that is mentioned in your second unit is the enzymes. So once we